We'll get started in a couple of minutes if that's okay, but yeah. Right, everyone, hello. Welcome to today's lesson. So today's session is on transport in animals, which is basically a lot about heart, capillaries. There's a lot of anatomy to it, and I'm going to try and explain the anatomy. If you bear with me, I've got a cool program that might be able to explain the anatomy slightly cooler in a bit more of a 3D way. So I'll try and give you that a little bit. I'm going to just quickly admit some more people as they join. So we're going to start by talking about the structure and function of blood vessels. So you've got blood vessels. So you've got three main types of blood vessel. You've got arteries, you've got veins, and you've got your capillaries. And we're going to talk about the structure and function of all of these. And we're going to talk about them using cross-sectional diagrams. So what I'm going to do is I'm going to try and get you, while I'll show you a bit of the slides, I'm also going to show you through a little bit of 3D anatomy to it, just so that you feel more comfortable about the anatomy, because it often can be quite complex to try and imagine and visualize what this actually looks like. While that's going on, while it's loading, I will just explain kind of the learning outcomes. So we'll talk about the structure of blood vessels. So your arteries are made up of kind of all of your blood vessels are made up of three layers. So you have your tunica intima. Your intima is your inner layer. The way I remember that is intima, I-N, intima, inner. The other way you can think about it is intima. The actual root word of that is intimate. One of the root words of it is intimate, and intimate is something that's quite close and nearby. So it's intima to the blood flow. Your middle layer is your tunica media, which is your middle kind of It's probably your thickest layer in your arteries. And its function, it's going to contain all your smooth muscle. It's there for the vasoconstriction and the vasodilation that's really important in arteries. And your outer layer, your tunica externa, is basically your communicative layer. It's your layer that is able to communicate with the other structures that are found within the artery itself. Sorry, I'm just wrapping up with this just so I can show you the model. Models, blood vessels, perfect. Here we go. Oh, let's go. Can you see the models? Let me know if you can't. So I'm going to start with this kind of. We'll start over here in the arteries. We'll zoom in a little bit. So as you can see, your arteries has your 3 distinct layers to them. So your tunica intima is very thin. It's this kind of inner layer. And if you kind of look at that a little bit more in more depth, it's a very thin layer, and its function, basically, it's made up of 3 different structures. It's made up of your vascular endothelium and then an internal elastic membrane. So your internal elastic membrane that's found in your tunica intima, its function is for elasticity. It's going to contain lots of elastin, which allows for stretch and recoil. And if you imagine, that's quite important in an artery, because an artery often has blood flowing at incredibly high pressure through it, which means that it's got to have some stretch and recoil to it. If you imagine like a pipe, because the way I like to think about it is I like to think about your blood vessels are kind of like your plumbing work inside. They're the pipe work where everything is flowing, the blood is flowing through it. And if you imagine that, your artery is kind of like a pipe, if it was rigid and it had blood flowing at incredibly high pressure, it would not be able to respond to that. It would literally burst. So in order to withstand some very high pressures going through your arteries, your internal elastic membrane contains your elastin structures, which then are helped with resistance. Your endothelium, your vascular endothelium, is like any endothelium that you find in your body. It's kind of a single layer of endothelial cells. And again, this forms a sheet around the artery. And you can see that your vascular endothelium is incredibly tight. It's got a barrier. And the idea is it literally is a barrier. The idea is it's to stop anything from getting out of the blood. So it's got junctions between the cells that are incredibly tight, and they're there to stop anything from going outside the cell. So that's your tunica intima. So if you remember, intima is your internal elastic membrane and your vascular endothelium. If that's all you remember in today's lesson, from this bit, from your tunica intima, that's fine. The next layer is your tunica media. And I just want to show you, I want to highlight this. This is really important for you guys to know. Look at the difference between the tunica medias of your arteries and your tunica media of your veins. You can clearly see a distinct histological difference in the sense that the tunica media of the artery is incredibly thick compared. And this is because of this incredibly thick layer of smooth muscle. And this allows for vasodilation and vasoconstriction. This basically means that the volume of blood that can be passed through an artery in any given time is variable. You can have, if you're in periods of high pressure, you can vasodilate and you can have more blood flowing through at lower pressures, so it can help lower the pressure. And if you have lots and lots of blood flowing through, then if you have a very small amount of blood, you can vasoconstrict, you can increase the pressure. Now, I want you to think about something quite logical from your baseline knowledge. What is the function of an artery? Because there are two functions. It's to take blood away from the heart, but there's a secondary function. I'll give you a second to think about this. This is really important for you guys to consider. What does anyone think of the secondary function of an artery? Anyone, if you want to put it in the chat, feel free to. See if you can get some brownie points for getting this correct. Let's get some interaction going. Carrying oxygenated blood? Not necessarily. Carrying oxygenated blood is something very important. It's the second one. Yeah, everyone's sort of saying this. Maintaining the control of blood pressure. It does kind of do that. But it also has the secondary function of, you'll notice that arteries feed into arterioles, which feed into capillaries, which means although it also maintains the control of blood pressure, if you had blood pressure going at the pressure that it was going in through an artery, or the blood pressure that it came out of the heart at, and it went through a capillary at that blood pressure, the capillary would be done. The capillary would burst instantly. So its secondary function, arteries and arterioles have the secondary function of lowering the blood pressure. Their job is to lower the blood pressure enough so that it can safely pass through capillaries without bursting them, but also will allow sufficient time for the exchange process to happen at the fenestrations within the capillary walls. And that's really important for you guys to note, because it is something that does, it will help you kind of visualize thinking about how this kind of works. Yeah. And then your last outer layer is your tunica externa. So your tunica externa has an external elastic membrane, again, more elastin fiber for stretch and recoil, but also has the vessels that the artery supplies the artery walls with. And that's really important to note. So if I was to remember kind of a summary of arteries carry blood away from the heart to the rest of the body. They're thick-walled to withstand high blood pressures. They contain elastic tissue, which allow them to stretch and recoil for smoother blood flow. They contain smooth muscle, which enables them to vary blood flow by contracting and relaxing and controlling lumen diameter. And they're lined with a smooth endothelium to reduce friction and to ease flow of blood. So I'm gonna put this in a sort of a whiteboard for you, and I'll switch quite quickly between the whiteboards and the so all of this has been taken from my own personal notes that I used to revise my A-level biology with many years ago. So arteries, these are kind of in your green. Remember, these are your artery function. I'll add to this whiteboard as we go along. Okay, so after we've got an artery, we move to an arteriole. So an arteriole structurally is very similar. They're a branch off of the arteries, but they're thinner and have less muscular wall, and their job is to feed blood into your capillaries. And I've put my arterioles in, oh, that's not a good color. You copy that down. Put them in purple. Next, capillaries. So capillaries are really, really important because they're the sites of predominant oxygen exchange with the environment. So capillaries are where you are basically exchanging your material with the external environment. So you need to be adapted for that. Now, let's all think back to our structure of alveoli and our structure of gas exchange. And we talked about 3 things that make gas exchange really efficient. And your teacher will have talked to you about those things. What would your teacher have said is the things that make gas exchange very efficient? Thin membrane, large surface area. Perfect. Thin membrane and large surface area. Abby, sorry, I've got a question here from Abby. Could I please slow down and give you a chance to write things down? Yeah, so all of the notes I will come back to once I've sort of filled that PowerPoint, that whiteboard, with all of the important information about arteries, veins, and capillaries. And we'll talk about all of that, and I'll give you a chance to write all of that down together. It just makes more sense than me kind of going through it. Yeah. 1 cell thick. So short diffusion distance, thin membranes, and a large surface area. Those are your kind of 3 main things that are going to increase your rate of diffusion. And this is kind of a capillary is adapted for that. So capillaries have very small 1 cell thick walls, and 1 cell thick walls are going to increase your, they're going to ensure a short diffusion distance. And these walls have fenestrations in them. So remember, oxygen needs to get out, and oxygen can undergo simple diffusion, but there are other molecules that need to get out to the cells. For example, amino acids. Cells need amino acids so they can grow and they can form proteins. Cells need glucose. Glucose is really important as their principal source of energy, or a principal metabolic substrate for respiration. So they need both of those things. So those can't get out through simple diffusion alone. And your capillary cells don't have the necessary ion channels, and they don't have the necessary electrolyte balance to start doing movement through electrolytes. So what they're going to do is they're going to use fenestration. So fenestrations are thin structures in the walls of the capillaries, and they allow for things to kind of pass out through them, for larger molecules to pass out through them. The next thing that you would want to think about is capillaries are, so they're the site of metabolic exchange, they're 1 cell thick, and they've got fenestrations. You can also see that there are lots and lots of branching capillaries. So capillaries branch off into a network of capillaries. So capillaries usually exist as a capillary network. And this capillary network increases the surface area for exchange to happen. So if you think about 3 things that capillaries do to make themselves more efficient for gas exchange: 1 cell thick walls, fenestrations, and networks to increase their surface area. So I'm going to add that, we can add this now to our our whiteboard of information about the different structures. And what I can also add to this is they also exist in large branching networks to allow for an increased surface area for efficient gas exchange. Right. So those are your DI. What is the title for today's lesson? Today's lesson is titled Transport in Animals. So we're talking about how animals transport things around their bodies. So we're going to be talking about the heart. We're going to be talking about the cardiac cycle. We're going to be talking about ECGs. We're going to be talking about coronary artery disease. We're going to be talking about all those things that are related to kind of the structure of how things work and how things are brought around the body. Now, veins. So you've now got your veins, and veins are bringing things back to the heart. Now, what you can imagine is, you can imagine the blood. The blood has traveled a significant distance by this point. If you imagine like you running a marathon or running a cross-country sprint, you start out really quickly, you know, you're racing through this, but by the time you're getting back, you're tired, you're slow, and that's kind of the way that blood is flowing through your veins. Your veins, the blood is flowing slowly. It's not got a lot of pressure to it, which means that the function of the vein is to bring as much blood back as quickly, but also to stop blood flowing the other direction. If you imagine that there are veins in your legs, right, that are having to work against gravity as well. So it's very challenging for veins to bring blood back to the heart. So if you imagine what they're doing is they don't need to control their luminal diameter because the pressure is fairly constant through a vein, which means that they don't need to increase or decrease their blood pressure to manage different circumstances because the blood is fairly constantly at low pressure. But that's why they can get away with having thin tunica medias and even thinner internal and external elastic membranes because they don't need a lot of that. The one thing they do need is valves. Valves are one-way unidirectional structures that basically prevent blood flowing back through it, through the other side. The way I would think about it, or the way I would imagine a valve, is it's like, it's kind of like a postman's flap. You can go in one way. The postman can get his, the letters through the door, but he's not going to get them out. They're not going to go the other way, and they're not going to come out of the door. So valves functionally are designed to prevent backflow of blood through their structures, which means that blood can only flow in one direction, which is really important because otherwise you get a lot of problems. And you get basically, if there were no valves, blood would pool at the bottom, it's in your feet, and then that would make, if blood is pooling, very low pressure, your legs would start swelling. And if your legs start swelling, you'd have buildup, you'd have oxygen issues, and that would be a much bigger kind of issue. So veins, venules and veins. I'm going to add them we can now add those to our whiteboard. And that's kind of it. I'm hoping that from this kind of little bit of the first bit of the talk, you've been able to appreciate just going to quickly copy and paste. Oh, no, veins. We've got in blue. And so we've now added and we've kind of completed our sort of summary whiteboard with the key information that you need. So I'm just going to quickly add this little bit about arteries. Green. Move this a little bit up. Down. Hopefully, let me know if there are any issues with you being able to see that. I'm going to give you a minute or two to write this down. In the meantime, I'm going to go back through the chat and answer any questions. So, which website is this? So this is Complete Anatomy. Again, I wouldn't recommend it for you. You guys don't need to know a lot of anatomy for your A-levels. I basically used it for med school. It was very useful at that point, but it's probably not very useful at your stage in your education. Search up Complete Anatomy. Yeah. Do I record these lessons every week? Yeah. So these lessons will always remain recorded, always remain free, and you can always watch them back on YouTube at any time. And again, they'll forever remain accessible to people. What does no pulse of pressure mean? So pulse is basically a bounding pressure that you get from basically the stretch and recoil of an artery. So if I go in and I feel my radial pulse, which is here, you should be able to feel your radial pulse. Yeah, all of you're probably able to feel your radial pulse. That's basically the blood's flowing through it. It's at high pressure. So stretch, recoil, stretch, recoil, stretch, recoil, stretch, recoil. And that's what pulse is. Basically, in a vein, you don't get pulse because there don't need to be stretch and recoil because the pressure is so low. So what I mean by pulse is I mean pulse pressure, which is basically the idea that there is no kind of pulse pressure. Would I only need to know the structure and function of the blood vessels? No, there will be more. this lesson isn't over at 26 past, but there is a lot more to learn for this topic. But I'm giving some time to kind of digest that content. What's the difference between arterioles and capillaries? So an arteriole is a branch of an artery, still going to have thicker walls than a capillary. An arteriole isn't going to be one cell thick. And the function of an arteriole is primarily to reduce the blood pressure. So it's going to have very turbulent flow to reduce the blood pressure. Again, if you imagine the arteriole, it's kind of like the brake on your in the blood pressure. So you're cycling down a hill, you've got lots and lots of speed going at you, you're speeding very, very fast. The arterioles is going to have really turbulent flow to brake your blood pressure and to reduce it. So the capillary has no carrier or channel proteins for large substances to diffuse in. I'm not going to go as far as saying no, because there will be the basic carrier and channel proteins for it to fulfill its function, but it's got no specialized SGLT2 channel proteins for glucose transport. What is the order in which the blood passes through these blood vessels? So heart, artery, arteriole, capillary, vein, venule, heart. Sorry, no, wrong way around. Artery, arteriole, capillary, venule, vein, heart flow. Are the recordings for these lessons on YouTube? Yep, they will always be on YouTube. And what's the name of the YouTube channel? AQA Study Mind, pretty sure. If you just Google Study Mind, it'll be on there. Perfect. Right. That's kind of us having talked about this. We've talked about all of these, but this is a very good summary table, which I do recommend kind of you getting down into your notes as well. Just it's a summarization of everything that we've talked about so far. Again, just if you take a picture of it or some description. We're not really going to talk about this because this isn't accessible to all specs. Okay, the next thing we're going to talk about is we're going to talk about a little bit about the exchange in capillaries. So by this, we're talking about tissue fluid. So tissue fluid is a substance. Basically, not every cell in your body is going to have a capillary right next to it, right? We can imagine that. That's quite logical. Not every individual cell is going to have a capillary right next to it, but every individual cell is going to need nutrients. It's going to need oxygen. It's going to need glucose. It's going to need the amino acids to build proteins. It's going to need fatty acids to build lipids. Every cell is going to need that as an individual cell, but not every cell is going to have a capillary next to it. So we need some way of getting all of these substances from within your capillaries, from coming outside of your capillaries, and then we need something to get them to those individual cells. And how do we do that? We do that through tissue fluid. And tissue fluid is this substance. It's a form of plasma. So plasma is going to have everything in it. Plasma is the fluid that carries, basically it's the fluid that forms blood. So plasma is going to contain all of your nutrients, but it's also going to contain your red blood cells, your white blood cells, your lymphocytes, your platelets, things that individual cells don't need to have. Individual cells don't need to have red blood cells with them. And tissue fluid is basically a way of, it's a form of plasma. It's going to contain the same fluid, but it's not going to have the cellular component to it. And how is tissue fluid formed? So there are two ways to understand this. There are two pressures that you need to understand. There's the hydrostatic pressure. So hydrostatic pressure is pressure, if you imagine that this is your capillary. You have the arterial end, and you have the ventral end. The hydrostatic pressure is the pressure that is pushing this fluid against the walls of the capillary. And this is pressure that has been generated by the heart when it pumps. Hydrostatic, if you break that word down into its constituents, hydro meaning water, but in this case, we're going to talk about fluid. Stasis meaning movement. So hydrostatic is the pressure from the movement of water or the movement of fluid. So hydrostatic pressure is the pressure generated from the movement of this fluid. And you have the hydrostatic pressure pushing against the walls of these arteries. Your oncotic pressure is your pressure from the fluid outside the arteries pushing on the wall of the artery or of the capillary. So your oncotic pressure is that pressure that is coming from the fluid that has already been released from the capillary, pushing back down on the capillary. And tissue fluid is formed by a mismatch of these pressures. So at the arterial end, the hydrostatic pressure is greater than the oncotic pressure, so fluid moves out of the capillary. But now you have a lot more fluid out of the capillary than you do inside the capillary. So at the ventral end, the oncotic pressure outweighs the hydrostatic pressure, so fluid moves back into the capillary. And this net difference between the pressures is called the osmotic pressure. So the osmotic pressure is the difference between the hydrostatic and the oncotic pressures. So hydrostatic pressure, osmotic pressure here at the arterial end is negative because there's more fluid going out. So there's a net negative fluid. But at the oncotic, at the ventral end, because there is more pressure pushing in than pressure pushing out, the osmotic pressure is positive. The problem is, though, if we think about what's in tissue fluid and we think about the constituents of tissue fluid, we have small proteins, and we have lots and lots of ions, and we have lots and lots of things that not all of which are going to be taken up by the cells, which means that we have two fluids of not equal protein pressure, which means that even at this ventral end, not all of the residual fluid that has not been taken up is pulled back in, and there is some fluid left over. So there is net fluid left over. Don't worry about taking too many notes on this. Again, I'm going to give you a whiteboard of core information you need to know about this. But there's net fluid left over, and this net fluid we now call lymph. And lymph is reabsorbed into your lymphatic system, which is a blind-ended system. And your lymphatic system carries this remaining tissue fluid, which has not been pushed back into the capillaries. And it also contains lymph nodes, and the lymphatic system forms a key part of your immune system because it helps remove bacteria and other pathogens. But again, I'm going to give you a whiteboard of information on this. So don't worry about taking too much information down right now. So this is a lot of information, I know, but I will make it in a way that kind of makes sense. So I'm going to restructure this a little bit. I'll start with just a little bit on tissue fluid and the constituents of tissue fluid, and then I will do the lymphatic system slightly later on again. So and this is kind of a little bit of a summary table about the constituents of the different structures. So this is blood plasma. So again, I'll give you some time to write this down. And while I'm doing that, I'm going to just quickly send out a review link if possible, just so if you are able to give a review and some feedback on this session, it would be very helpful to me, purely because it will help me see kind of what you've enjoyed about these lessons, what you think. What your thoughts are, and I'll send a link into the chat with that. Again, it will take a couple of minutes just to do. That's okay. So I've sent a link in the chat with some review link. Again, if oh sorry, I've only sent that to one person. There you go. Send it to everyone. If you're able to fill that form out, again, that would be really helpful. Would give us a lot of indication about how helpful you're finding these sessions and if they're helping you with your revision. And if you can fill this out, we'll also be giving away 1 hour of free one-to-one tutoring to one person who does fill it out. So if you fill it out, put your email in the chat, and then while before this is all uploaded, it will kind of go through a process, and we'll be giving out 1 free hour of one-to-one tutoring as well, so you can get the best. I've been asked a question: what are thrombocytes? So thrombocytes are cells that produce platelets. Platelets are helped in the blood clotting process. Would you like us to reference your name? If possible, yeah, my name's Hamza. I'll put that in the chat. If you can reference my name, that would be great. It would be helpful to just kind of show people my tutoring style. Again, that'd be quite useful. So I hope that answers your question. Thrombocytes are cells that produce platelets. If there are any other questions at this moment about this bit of the content, again, we do still have quite a bit to go through. We'll be talking a little bit about heart anatomy. Ideally, I will also want to get through hemoglobin, the oxygen saturation curves. I'm hoping we'll have time to do all of that today. Again, it is difficult trying to run through what is essentially 5 weeks worth of work in 1 hour. But again, I'm trying to give you a summary of everything your teachers will be saying. Is the fluid in equilibrium, equal amount moving in, equal amount moving out? No, that's why there's residual fluid called lymph. Because if it was equal amount moving in, equal amount moving out, no, it's to do with equal pressures moving in and equal pressures moving out. But because the pressure that's generated by the proteins is dependent on the amount of proteins that are in each fluid, that then becomes different. It's not about amounts, it's about pressures. Will we receive the recording via email? Good question. No, the recordings are all on YouTube. I will also show you how to access those kind of later on in today's session, just for people for future reference that, you know, might not be able to, if you're not able to access that. Right. I will give you until about maybe 44 past. No, I'll do 43 past. So 2 more minutes just to take some notes down. Sorry about that. Because I do kind of want to get back to the anatomy. I've got a little bit more cool anatomy to show you. If you guys put your emails in the chat after you've done the review process, again, we'll be giving out 1 free hour tutoring. So you can, if you're able to do that, that'd be great. Right. Back to some more content. Thank you for submitting the review form. Proven very helpful. We're not going to talk about this. We're actually going to go to this then. So, the human heart. So, structure of the heart. Let's talk about that first. So, let's discuss this. So, the heart is a 4-chambered organ. Is this year 2 content? I've been asked. And this is year 1 and year 2 content. I mean, in the sense that it'll come up in your A2, but it is year 1 content for a lot of specs, depending on the spec that you are picking. Let's reflect that. Sorry, I'm doing some dissection stuff here. Reflecting that back. Okay, so what I'm going to do is I've reflected the outer wall of the heart, just so you can kind of just so I can go in a little bit and see what's going on in here. Basically, the heart is a 4-chambered organ. So we go in, zoom all the way into the heart. We might not be able to get all the way in, but if we kind of look at this heart from a gross structure, you've got basically an atrium here. So this is an atrium. Is this the left or the right atrium? KM, is this applicable to the OCRA example? This has been taken from the OCRA example. So yes, all of this is applicable to the OCRA report. Right atrium. Perfect. Now you've got the right ventricle here. Then on the other side, you've got the left atrium and the left ventricle. So it's a double circulatory system. Humans have a double circulatory system, which means that the blood travels through the heart twice on each circle and each loop of the body. Basically, this is to do with the idea that it prevents mixing of oxygenated and deoxygenated blood. So if I talk you through the process of when blood enters the heart. So blood enters the heart through the vena cava, which is this structure here. It enters into the right atrium. It then passes through the right ventricle and out through the pulmonary artery, which you can see coming here, left and right pulmonary arteries. It then re-enters the body through the pulmonary veins, which are these structures, into the left atrium, passes into the left ventricle, and then out through the aorta. Unfortunately, I can't give you a whiteboard bit for this because this wasn't in my notes from many years ago, but I can write it out for you. So 1, vena cava, 2, right atrium, 3, right ventricle, 4, pulmonary artery. And the way you remember artery is A is away, so arteries go away from the heart, and pulmonary vein, the last 2 letters is literally in. 8, aorta. So one thing that's really important for you guys to note and learn is the left ventricle is thicker than the right ventricle for the fact that it has to transport blood a further distance. So if you imagine that, if you look at your body, your heart and your lungs fairly near each other, but your aorta has to go all the way down to even your legs. So the blood pressure that it has to generate, the left ventricle has to generate much higher than the blood pressure generated from the right ventricle, which means the blood has to travel a further distance, which means that it is going to be, it's much more important that the left ventricle is thicker. Now, that's your baseline anatomy. Once we've done that, we've talked about that. Let's talk about the electrical conductivity within these hearts. So quickly talk about the valves, actually. The valves, there are 4 valves in the heart. So you have your atrioventricular valve. So these are your 2 your left and your right atrioventricular valves. And they are between your atria and your ventricles. And atrioventricular valves are semilunar valves, which means that they look kind of like crescent moon. And basically, they're joined by chordae tendineae. So there are tendinous cords that attach those valve flaps to the walls of the ventricle. And then you've got your tricuspid valves, which are between your right atrium and right ventricle. So not right atrium and right ventricle. Your tricuspid valves are here between the arteries. So your tricuspid valve is between the right ventricle and the pulmonary arteries. And then your aortic valve is between the left ventricle and the aorta. So again, it is a closed double circulatory system. So double because it covers, the blood comes in through twice. There's a pulmonary circulation and a systemic. And closed means that the blood is only found in the blood vessels. And we know that. We've talked about this before. Okay, cardiac cycle, really, really important to know. So the cardiac cycle is basically contraction, contraction, relaxation. So heart sounds, the function of the cardiac cycle is to ensure that blood is continuously pumped around the body. And there are 2 heart sounds that you have to pick up on, lub and dub. So lub is your first heart sound, and it's when the AV valves close. And dub is the second heart sound, and it's when your semilunar valves close. So cardiac diastole. So diastole is relaxation. Whenever you see the word diastole, you need to know that that's relaxation. And the atria and the ventricles relax. So what does that do? Well, relaxation increases volume. Is this A-level or GCSE? This is A-level content. So diastole increases volume. And when you increase volume, by default, what are you doing? Decreasing pressure. So relaxation of the atria and ventricles increases the volume and decreases the pressure. This pulls blood in from your vena cava and your pulmonary vein into the atria and the ventricles. That's diastole. Now, atrial systole is the atria contracting, and atrial contraction increases pressure in the atria by decreasing volume. Once the pressure in the atria is greater than the pressure in the ventricles, the valves open and the blood flows from the atria into the ventricles. Cool, with me so far? And then ventricular systole is where the ventricle atria relax, so the atria pull blood back in, the ventricles contract, which means they decrease pressure. They increase pressure, by decreasing volume, which means that the blood flows out of the arteries and out to systemic circulation. And I'm going to give you a whiteboard for this because the cardiac cycle is really important to know. So you'll get a quick whiteboard for this. And I think we'll probably end on the note of the electrical conductivity of the heart. So sinoatrial node, atrioventricular node. And I will quickly put in at the bottom of this that this process is regulated by increasing and decreasing volume. Systole equals increase in pressure by decreasing volume. Diastole is a decrease in pressure by increasing volume. Okay, and then the last thing I'll quickly talk about, and I'll give you a whiteboard on as well, is myogenic conductivity of the heart and how electrical conductivity works in the heart. But I'll give you 2 minutes to write this down. Then we'll spend the last 5 minutes quickly talking through that. Oh, I've been asked a question. A-level, yeah, this is A-level. Am I a university student? Yeah, I'm a third-year medical student. Okay, last question is about myogenic conductivity, and we'll quickly touch on that. So this is kind of really important because it describes why the atria contract differently to where the ventricles contract. And we can link this to ECGs. So myogenic basically means that the heart can initiate its own contraction. In the walls of the right atrium, there is a region of specialized muscle fibers called the sinoatrial node. And they initiate a wave of depolarization that span across the atria of the heart. This this signal is detected by the atrioventricular node. So there is a septum between the atria and the ventricles, which basically prevents electrical conductivity from going down just from the atria straight to the ventricles. The AVN initiates a delay. So when I draw out an ECG structure, which again is something you should know for your A-level exams. So when I draw out a structure of an ECG, let's say we've got P. That's bad. That's a bad one. So that's your general structure of an ECG. So this P wave is atrial depolarization, or atrial. It corresponds to atrial systole in the cardiac cycle, but it's basically sinoatrial node stuff. So if you've got a problem with your P waves, you've got a problem with your sinoatrial node. So atrial depolarization is your P wave on your ECG, and it's SAN, and it corresponds to atrial systole. You can see that there's a small gap here between where the P wave ends and the next wave begins. And this is the delay in the AVN. So this is the delay in your atrioventricular node. Your next bit is your QRS complex, which represents, does anyone know? It represents ventricular depolarization. So if you have an issue with this wave, this could be anything from your AVN all the way down to your Purkinje fibers or your bundle of hairs. And it roughly corresponds to ventricular systole. This gap between the S and the T is something called a plateau phase, which you'll talk about way more in detail at university, but don't worry about it too much, but just consider it plateau phase. And your T wave is diastole. So this is relaxation. So if there's a problem with your T wave, there's a problem with relaxation, or the problem with relaxing the muscle fibers. What is the role of Purkinje fibers? Well, so remember, the AVN is kind of in the middle, but your ventricles need to contract all the way up. If you imagine this is the kind of a drawing of the heart, bad drawing, but the AVN is kind of down this middle bit. The Purkinje fibers need to ensure apical contraction. Basically, you contract from the bottom up to generate as much power as possible, is the idea. And it kind of works. Right. Sorry to be, today we won't have time for questions. It's been an intense 1 hour. Yeah, again, I will quickly show you how to access it on YouTube. So if you go onto your YouTube app, you go into StudyMind's YouTube page. Go into StudyMind. You will have all of our previous videos. So all of our previous live videos from all of our previous lectures, and even this one that will be from today. So all of them will be recorded directly onto YouTube. And yes, this was corresponding to the OCR spec. A little bit more advanced than the AQA spec. And AQA, you would need to know a little bit more about conditions, which again, I wasn't really talking about because I didn't have the time, unfortunately, for basic. I was focused on basic physiology. But thank you for your time, guys. I'll hope that was good.